Hello everyone, my name is Jen Lee, and today I will be presenting my project on the identification of stomatal-related tDNA mutants in Arabidopsis thaliana. Due to global climate change, drought is quickly becoming a prevalent problem in agriculture since it causes devastating crop loss. The crops, to minimize water loss, have structures called stomata, which consists of two guard cells and a pore in between, and this regulates gas exchange. The more stomata you have, the more water the plant loses. The less stomata you have, it actually increases plant survivability. So the development of stomata is regulated by a variety of genes, but we will be focusing on one in particular, EPF1. EPF1 binds to the receptors ERL1 and TMM, which then causes the activation of a MAPK cascade. This cascade degrades the transcription factor MUT, which prevents the stomata development pathway from proceeding. Thus, EPF1 acts as a negative regulator of stomata development. We know what happens downstream of EPF1 as demonstrated by this figure, but we don't know what happens upstream. In other words, how EPF1 is regulated. We know how EPF2, which is on the left, is regulated. The transcription factor it degrades, speechless, is its own regulator. Therefore, we plan to study the EPF1 promoter to figure out how it is regulated, as its regulators lie outside of this pathway. We will be confirming the insertion through PCR. We have designed primers specific to the plant lines that we will be using, including a mutant primer that will be able to replicate the gene with the insertion. If it cannot, then the plant is considered wild type. If it can, and there is no wild type band, then the plant is homozygous for the insertion. Example of this can be seen in the figure of the right. Now, the insertions themselves are just long sequences that are inserted into the genome of the plant and can disrupt the gene's expression, and there should be an observable effect on the expression of that gene if it has been successfully inserted. We will be using a total of 8 lines, all with a putative insertion. The insertions are either in the gene EPF1 or in the gene ERF53, a gene directly upstream of EPF1. This is a figure showing the relative locations of the putative insertions. All the insertions are located in the promoter region of the genes, except for SOC035606, which is located in an exon of ERF53. After conducting PCR using Columbia 0 as our control, we found that these lines were wild type, meaning that there was an insertion in those lines. We know this because only a band using the wild type primers, which is the top band, showed up, and not with the mutant primers, which should be on the bottom band. More interestingly, these lines were found to have at least one homozygous plant in each line. For example, plant 10 of SAL367D11 is homozygous as it has a band for the mutant primers, but not the wild type primers. We use these homozygous plants to test for their mRNA expression of EPF1 using Columbia 0 as our control. Genomic DNA as a size comparison, as EPF1 is, after mRNA processing, should be smaller than its genomic equivalent, and EIF4A as our housekeeping gene. We found that 2 of the lines demonstrated a decrease in EPF1 expression, while the other 2 lines demonstrated an increase in expression, demonstrating that the insertions in these lines did have a marketable effect on the expression compared to the control, which is the Columbia 0. The lines that were specifically affected concerning the decrease in EPF1 expression were SOC144857 and SOC023569. An increase in expression was found for SOC035606 and SAL367D11. However, it should be noted that 2 of the lines were inserted into the upstream gene ERF53, SOC035606 and SOC144857. The reason why we decided to test for ERF53 is that the promoter region of EPF1 is not actually well defined. ER53, again, is a gene directly adjacent to EPF1. It encodes a drought-induced transcription factor, and this transcription factor binds to promoters downstream. So because the promoter region is not well defined, there could be some overlap between the 3' end of ER53 and EPF1's promoter. Therefore, any alteration in the expression of ER53 can have also an effect on EPF1. So after conducting these experiments, we do have a solid control for further experiments. We plan to look at the phenotype of these lines under the confocal microscope. as well as analyze the interrupted promoter sequences to see if they serve as putative binding sites for any regulators, as well as grow these lines under drought conditions. Thank you so much for listening to my presentation. If you have any questions, please feel free to ask.